Good morning, respected professors, teachers, and distinguished guests. We are humbled. We are humbled by your presence. We have gathered here today to celebrate the National Science Day. National Science Day is celebrated on 28th of February every year to celebrate the study of Sir C.V. Raman, the Raman effect, for which he was accorded with the greatest prize of science for the benefit of mankind, that is a Nobel Prize. So we are here to celebrate not just only to honor Raman effect, but also to honor many scientists, researchers, from the likes of Sir Jagadish Chandra Bose to Sir Satyendranath Bose to Meghnad Shah till the greatest applied chemistry India has ever produced, Acharya Prafullo Chandra Ray. We are here also to celebrate many research scholars, scientists whose name has been not imprinted in the footprints of the sands of time. So I would like our monastic members to kindly begin the program by the holy Vedic chanting. Maharaj, please.
Harihi Om Sahana Bhavatu Sahano Bhunaktu Saha Viryam Karvavahai Tejasvinavadhitamastu ma Vidvishavahai. Om Shanti Shanti Shanti. Harihi Om Shanno Mitra Shamvarunaha Shanno Bhavatvaryama Shanna Indro Brihaspatihi Shanno Vishnurrukramaha Namo Brahmane Namaste Vayo Tvameva Pratyaksham Brahmasi Tvameva Pratyaksham Brahma Vadishyami Ritam Vadishyami Satyam Vadishyami Tanmāvatu Tadbhaktāramavatu Avatu Mam Avatu Bhaktāram. Om Shanti Shanti Shanti. Harihi Om. Om Bhadram Karnebhih Shrunuyama Devaha Bhadram Pashyemakshabhyajatraha Sthirairangai Stushtrubhagam Sastanubhihi Vyashema Devahitayadayuh Swasti na Indro Vriddhashravaha Swasti na Pusha Vishwavedaha Swasti nastarsho arishthanemihi Swasti no Brihaspati dadhatu. Om Shanti Shanti Shanti. Harihi Om. Purnamata Purnamidam Purnat Purnamudachyate Purnasya Purnamadaya Purnamevavashishyate. Om Shanti Shanti Shanti. Harihi Om.
Thank you, Maharaj, for the Vedic chanting. I would like to kindly ask our revered Swami Ekachitananda, our principal Maharaj, to kindly give the welcome address. Maharaj, please.
Om Namo Bhagavate Ramakrishnaya. Respected Professor Abhijit Saha, University of Jerusalem. Respected Professor Pratap Kumar Dash, distinguished faculty members of the Department of Industrial Chemistry, research scholars, students, and all the members of non-teaching staff who have joined this webinar. My respectful namaskar, love and good wishes and due places. So as our Saurabh had been mentioning, there's a celebration going on, and this industrial chemistry department enjoying, celebrating in their own way, because in this pandemic period in particular, they have contributed substantially to the fundamental research. And only the other day, the head of the department has been honored at the national level, and we feel so much proud of that. I'm sure that his original contribution, along with the substantial support of all the faculty members, all the guest lecturers, and in particular today's webinar, is going to contribute a long way in the development of the department. And I pray to the Holy Trio for the successful completion of the webinar. Om Shanti 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 Hari Om Tat Sat Shri Ram Krishnarpanamastu.
Thank you, Maharaj, for the warm and the cordial welcome. And so, the man who has worked in the protein science for long, the decade should be not numbered off. It would be very small in front of you. He is a distinguished alumni of our college, Ramakrishna Mission Vidyamandira. So, so I would like to kindly ask Professor Pratap Kumar Das, former scientist of Indian Institute of Chemical Biology, to kindly give the keynote address. Sir, if you can kindly give.
Am I audible, Saurabh?
Yes, sir, you're audible. You're absolutely audible.
So good morning, everyone. Thank you, Saurabh. Thank you, Maharaj. Thank you, participants, speakers. Thank you all. I particularly thank the organizer, Saurabh, Uttam, et al., for arranging this wonderful webinar around an interesting area of research called the peptides and their applications, broadly speaking. I also thank the organizer, Uttam, Saurabh, etc., for giving me this opportunity to say a few words at the outset about the webinar topic in general, about the speakers as far as I know their work, and about the talk expected to be delivered by them. Little bit of it I will just deliver. Peptides, as we all know, are polymers of amino acids. Simple. Unlike other biopolymers, particularly of carbohydrates, fats, etc., fatty acids, etc., peptides are an interesting class of molecules, macromolecules rather, in the sense that they offer interesting properties as a material. They are malleable, they are stretchable, they are compressible as well. Likewise, they are relatively with relatively higher density as compared to, say, carbohydrate polymers or fatty acid polymers. They also are capable of imparting desired electrical or electronic properties through design synthesis. Perhaps the theme of this topic speaker's talk today will center around these things, how they will design, synthesize many useful peptides for some useful purpose. That's the game of applications. So no wonder, therefore, my dear students, you can design, synthesize a polymeric material of your choice for the binding with your desired target, for the binding to your desired target, be it slightly more cationic or anionic or whatever way you like, polar or non-polar. You can design synthesize that. So you can build your peptide around your desire. Peptides, broadly speaking, have many applications, and the perspective is quite large. The field applications in the biomedical science field is quite vast. Many of you, the students, I'm addressing to students mainly. Many of you may have already heard of few terms, including peptide, which includes the term peptides, like peptide vaccines, cyclic peptides, antimicrobial peptides, anti-cancer peptides. Regarding peptide vaccine, there are quite a few cancer vaccines coming up in recent times. You may be knowing that glioma cancer, for example, is coming up for use of as peptide vaccines for treatment of glioma cancers. One can use recombinantly expressed antigens. Commonly we call it subunit vaccines. An area of extreme interest around this concept is the application of nanotechnology. To vaccine development, which allows the antigens to be expressed on a particularly delivery system. One of the most exciting examples of nano vaccines are basically what we call rationally designed peptide nanoparticles. Simply speaking, rationally designed peptide nanoparticles. Remember, such nanoparticles never misses to follow the basic essence of science, physics, chemistry, etc., like rules of structural biology, biophysical chemistry, never misses the basic tenets of rules of biophysical chemistry, and also vaccinology. It fulfills all such criteria while getting changed along this synthesis design platform. So one can develop, in principle, in theory, a safe, protective, and easily manufacturable vaccine, which is a peptide vaccine, which can be a peptide vaccine. Regarding antimicrobial peptide, we have been hearing about this for quite some time, which is another important area of applications related research that are coming up in recent times. Antimicrobial peptides, you all know, are an important component of innate immunity, unlike adaptive immunity. Thus, in this failure world of antibiotic usage, microbial resistance, etc., design and prospective application of antimicrobial peptide is of the order today, as antibacterial or antifungal, antiviral, or even anti-biofilm formation. Such immune modulating peptides are of current area of interest. This is another area of applications. We call it antimicrobial peptides. Further, one can think of, just not think of, it is being practiced nowadays, that peptides with anti-cancer activity. Currently, we see lots of efforts in working around anti-cancer activity of natural as well as synthetic, semi-synthetic peptides. Many examples are coming up in recent times. I can name more, but one more I should say that the peptide is known for its application is what you call cyclic peptide. A cyclic peptide, interconnected. This cyclic peptide, both natural and synthetic version are available. They are being used as biochemical tools, biological, chemical, different experimental requirements, biochemical tools. As for example, we may call it ionophores to study structure functions, ionophores to study structure functions of membranes and transports, membranes and transports across it. Coming to today's webinar, we have two very good young speakers who will share their excitements and views about two important and rather pertinent issues of peptides to be able to make it applicable. They are doing hardcore chemistry, but very pertinent points they are nurturing around. So they are Dr. Abhijit Saha. In the first session, he will speak on, and in the second session, we will have Dr. Kingshuk Basu. Regarding Dr. Abhijit Saha's talk, what I could gather is he has designed peptides through various experiments. It could be dry lab experiments, it could be wet lab experiments, to prove points like how to design specific peptides that can target a specific receptor of a, say, cancer cell, particular cancer type cells, without hampering the normal cell surface receptors, without interacting with normal cell surface receptor. That is perhaps one of the major goals. So he will be presenting his work, describing around his design planning, evidence for specificity, proof of delivery of the anti-cancer drugs using specific peptides of his choice, of his make, for specific targeting, So kind of what he call in Bengali maybe Arjuner Lokkhoved. That is his goal, Arjuner Lokkhoved type. Vis-a-vis the evidence that such targeting works with high specificity and minimal binding with normal cell surface receptors. Dr. Saha will definitely provide further evidence that such way of drug cells in the end. Perhaps if delivered to would understand. The other speaker in the second session, I give a brief outline about his talk simply to end my presentations, is Dr. Kingshuk Basu. He is another young, enthusiastic researcher I know who will address two very fundamental issues of peptide chemistry, basically. The nature and properties, the pattern, nature and properties of self-assembly and self-replication. These are the properties endowed with living systems. So he will be studying the component of living systems called, say, protein or peptides for this kind of properties. That is extremely important and issue to venture around, to look into the hows and whys of such issues. Self-assembly is a term which has a very deep-rooted meaning. A peptide can self-assemble, not just randomly, but in a programmed manner, creating a superstructure, macroscopic structure of a very different attribute, with a very different attributes of different physicochemical properties that can be explored and exploited. That can be explored and exploited in the field of applications in biomedical sciences. Now, such self-assembly characteristics can be augmented, reduced by changing 1 to amino acids in the core peptides. That's the way of their approach. Thus, one can look for opportunities. You students, please listen, that you can look for opportunities, at least theoretically, to create, say, chirality or to block the chirality purposefully, to perturb the existing chirality so that the process of self-assembly gets hampered or augmented. To look into these prospects, Dr. Kingshuk Basu wants to look into the macroscopic behavior, vis-a-vis the macroscopic behavior like drug release pattern. One such important endpoint is drug release pattern from such altered macromolecular assembly. From such altered macromolecular assembly. He has a keen interest to see how such patterns may have beneficial or deleterious effects on the efficacy of targeted drug delivery. Basically, his interest is around this, how self-assembly affects towards the benefit of different peptide for their applications. One such could be chemistry-wise, pi electron stacking in some regions to create organic, yet say electronic, I would say organic electronic material, to see how they are becoming advantageous, towards achieving different intended goals. You are trying to achieve some intended goals. Photoresponsiveness could be another such attributes one can desire to look into. Dr. Vasu will also touch upon the second important keywords called self-replication. That's the characteristic of life itself. So part of the cells, a living cell, which component is capable of making self-replications, we now know that peptide is the main player. Proteins is the main player. So many small peptides are also playing their roles. So that important aspect you want to probe with some design thinking like that. No wonder that such automation features are the vital attributes of any living systems. Peptides as an essential contributors towards that goal, to this vital goal, would therefore be an obvious choice to look into the why, how aspects of self-replications, how it is being happened. I hope this webinar will trigger young minds. That's my job, that's my desire, of the biology, chemistry, and even computer science students who are interested in drug design type of approaches, in more than one way, in many ways, in fact. Their appetite to know more through investigations will hopefully get stimulated to some extent from today's deliberations by two young scientists. So, with this brief remarks, I now hand over the mic to Saurabh for continuation of the webinar. Thank you all.
Thank you, sir. Thanks a lot for the keynote address. Thanks a lot. We are enlightened in the field of peptides. Perhaps we have had joyride on the beta plane and the alpha sheet. And so I would like our head of the department, Dr. Uttam Kumar Ghorai, to kindly give the introduction of the speaker. Sir, if you may.
Yeah. Thank you, Saurabh. Namaskar and good morning. Dr. Abhijit Saha is an ex-student of Ramakrishna Mission. He graduated in chemistry honors from Ramakrishna Mission Vivekananda Centenary College, Rohada in 2009. He completed his MSc in chemistry from Rajabhaja Science College, University of Calcutta in 2011. He qualified examination in chemistry IICB for PhD. He got the doctorate degree in chemistry in 2016. He also qualified the GATE examination in 2017. He worked at Hebrew University of Jerusalem, Israel for 3 years and 3 months as a postdoctoral research fellow. His research work is in the field of chemical biology. He has published 32 peer-reviewed international journal and hold 3 patents. So I would like to invite Dr. Abhijit Saha to deliver his talk on synthesis and use of peptide-based molecules for cancer treatment, targeted delivery of anti-cancer agent. Please, Dr. Saha.
Thank you, Uttam sir, for giving me the introduction about me, and thank you, Professor Pratap Das, for enlightening us with in good informations on peptides. Good morning to all, Honorable Principal Maharaj, my professors, my dear friends and students. I just want to tell you about my today's talk. Today's talk, I will discuss about how can we use the chemistry for treatment of cancer cells. Okay, I just going to share my slides first on. আমার স্লাইডগুলো কি দেখা যাচ্ছে?
হ্যাঁ, দেখা যাচ্ছে স্যার।
Okay, so my point is okay, okay, শোনা যাচ্ছে, কথা শোনা যাচ্ছে, স্লাইড দেখা যাচ্ছে, দুটো দেখা যাচ্ছে তো?
হ্যাঁ 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 স্যার।
Okay, my topic of my today's presentation is the synthesis and use of peptide-based molecule for cancer treatment. And can I use chemistry? Since I am from chemistry background, I use chemistry to make molecules. That is the synthesis part of my research, and I use the molecules for targeted delivery purpose to cancer cells. Okay, so before start for students, I just want to tell you about cancers and tumor. Actually, in our body, there are many normal cells. So I am pointing the cells, these are the normal cells. And sometimes some normal cells, due to some reasons, change their patterns and they're converted to cancer cells. Cancer cells are the cells who can divide much more than normal cells. In cancer cells, the properties of cancer cells are completely different from normal cells. So when some cells converted into cancer cells, they can divide much more than normal cells, and they have also property to make new blood vessels. That normal cells cannot make new blood vessels, but the cancer cells can have property to make new blood vessels. And during time, the cancer cells divide and divide and divide, and it form a lump. That lump we call tumor. And since they are making new blood vessels, therefore, when this lump is become very high, that small cancer cell can come through these blood vessels and can enter into the main blood vessel of body. As a result, if we have a cancer at one part of a body, that cancer cell can transfer to another part of a body. Because of this new blood vessels, since the blood vessels generated from cancer cell, the cancer cells come through the blood vessels and to enter to the main blood vessels, and it spread all over the body. Therefore, there are many treatments comes to treat this cancer disease. First, if I say the radiation therapy, in radiation therapy, people use high energy radiation like X-ray, gamma ray, high energy radiation to kill cancer cells. But there are some parts of body where we cannot give radiations. And therefore, some people use the chemotherapy. Some people use chemotherapy. In chemotherapy, people use the chemistry, the molecules. That molecules which can kill cancer cells, they just use that molecules as a drug to our body, and that molecule spread all over the body, and it goes to the cancer cells also and goes to the normal cells also. Since the probability to goes into the normal cells for the chemotherapeutic drug, so there are many side effects arise. Okay, many side effects arise. So because because the normal cells has some property, because this drug also kill the normal cells along with the cancer cells. So since normal cells are also killing, there are many side effects. So people try to think about the targeted therapy. Targeted therapy where they use the chemotherapeutic drugs to target to cancer cells so that it will not go to the normal cell. There are some recent two new therapy. There's hormone therapy and immunotherapy. These are the two new therapy. Okay. And surgery is one important part for cancer treatment. Bone marrow transplantation is another important part of cancer treatment. But today I will talk about this targeted therapy. That is my research during my PhD. Okay, first I will tell you about two molecules. One molecule is doxorubicin, another molecule is docetaxel. These are the two important drug molecules which are used for cancer treatment. First, doxorubicin, it is a hydrophilic molecule. And when we this molecule entered in our body, it interacts with our DNA. It damages the DNA, damage the DNA functions. Therefore, that cells is not able to divide. So, therefore, if this molecule goes to the cancer cell, it will kill the cancer cell. As well as if this molecule goes to a normal cell also, it can kill the normal cells. Similarly, docetaxel, it is a hydrophobic molecule. It interacts with the microtubule of the cell. So if this molecule goes to the cancer cell, it will kill the cancer cell. But if it goes to the normal cell, it will give side effects. So there are many side effects like hair loss and low blood cells, because it can also kill the blood cells also. Shortness of breath, vomiting, muscle pain. There are many side effects arise because of this chemotherapy. So people try to think how can we deliver those molecules specifically to cancer cell. For that purpose, one important part, we can look on the important part of cell line, that is the receptor, cell surface receptor. Okay, for each cells, for cancer cell, normal cell, for each cell, there are cell surface receptor on that cells. That receptor has important function for entering of the foreign molecule into the cell, inside of the cell. So if we have a drug that to enter into a cell, so drug can interact with the receptor and goes inside of the cell. So there are different kind of receptor for different purpose. Now, if we see the receptors are normal cell and cancer cell, we can see that in normal cells, there is also receptor. In cancer cells also, there will be some receptor. But since the cancer cells have divided much more compared to the normal cells, so cancer cell require much more kind of, if I say some more foods. Therefore, there are some more receptors on cancer cell, and some receptors are overexpressed. Overexpressed means there are some receptors, there are much more in cancer cell compared to the normal cells. So we can target this overexpressed receptor which are on that cancer cell, which we can target, because this overexpressed receptor means the more receptor on those cancer cells are present, but these receptors are not in the normal cells. Okay, therefore you are telling it the overexpressed receptor. Now I can tell you about some overexpressed receptor on the cancer cell. EphA2. EphA2 is a receptor that are overexpressed on cancer cell. P32 receptor, alpha 5 beta 1, alpha 2 beta 1, alpha 5 beta 3. These are the overexpressed receptors which are on the cancer cells. Now, since the receptor is a protein molecule and peptides is also part of a protein, so peptides have an ability to interact on this receptor. Okay, scientists have found some sequence that has an ability to interact with the receptor. Like if I say YSA sequence, this YSA, YPD, SVP, MMS. These are the code. These are the letter of the amino acids. Y means tyrosine, S serine, A alanine. Okay, these are the peptides. These peptides and interact with the targeted receptor, with the receptor EphA2. Similarly, CGNKRTR, this peptide can interact with the P32 receptor. So if we have this peptide, like CGNKRTR peptide, so this peptide will interact with the receptor of the P32 receptor. So if there is the cell of normal cell, it will not interact with the normal cell, because in normal cell, the P32 receptor is not overexpressed, but cancer it is overexpressed. That is the basics of my design of a drug molecule with peptide. So this as just for student, I just want to tell you that the amino acids when combined with the peptide bonds, amide bond, it forms peptide. When a peptide becomes large, it becomes protein. And these are some letters. That letters I used here, like YSA, YSA. These are the letters for the amino acid. Okay. Okay, now I am going to the design part, going to this slide where I can show you that this is P32 receptor and this is a cyclic peptide CGNKRTRGC. This is a cyclic peptide. This peptide interact with the P32 receptor, and after this interaction, this P32 receptor converted to NRP receptor, and this sequence CGNKRTR, this sequence enter into the cancer cell. This receptor is overexpressed on the breast MCF7 cancer cell line. Therefore, now I have to use this, and I want to deliver this cancer drug. That is, I choose docetaxel as a cancer drug to deliver to the cancer cell specifically with this NRP1 receptor. Therefore, I make a design of the CG. So I synthesize this CGNKRTR peptide. Okay, the peptide synthesis is very easy. I just showing you here. So for this peptide synthesis, I took a resin molecule. Resin is a solid. Resin is a solid molecule, polymer molecule. Okay, so this molecule has the functionality of NH2. For this, okay, now we have amino acids. Now in amino acid, the amine part is protected and acid part is open. So first I react with this NH2 with this acid part. Okay, then it forms the first amino acid attached with this molecule, and then I deprotect this the protecting group. Then amine part is again free like this, and again coupling the acid part with this amine. So like this way, I synthesize this peptide. That is cysteine, glycine, asparagine, lysine, arginine, tyrosine, and arginine. CGNKRTR. I synthesize this peptide. Okay. After this synthesis, okay. And now I will talk about the docetaxel. Okay. Before drug design, we have to know about the drug, because this is docetaxel drug. Okay. Now docetaxel drug, there are many important parts. Okay. This phenyl ring, this Bock protection. Okay. There are this OCH3 group, this one, this this 4-member ring. Okay, all this part have many importance for the activity of the drug molecule. So if I modify, okay, this part or this part or this part, so the drug activity will lost. Okay, so I found from many papers that this OH group has no functionality as such for drug effect on the cancer cell. So if I use this OH molecule for attachment of the peptide with this OH molecule, if I attach peptide with this OH molecule, it will not affect the drug function, because when drug molecule comes to a cancer cell, it interacts with also a receptor of the cancer cell, okay, or any cell. To interact with that receptor, the structure of the drug molecule, the structure or different part of the drug molecule is very important. So if we say this part, if we replace this phenyl part, it finds that the activity, this activity decrease. There is activity decrease, means the drug activity decrease. It will kill less cancer cells. So before attachment of a peptide molecule to a drug, we first have to find out where we can attach the peptide. Okay, so we have to find some functionality on a drug molecule which has access no function on drug effect. That part we can use for this functionalization. So I use this OH molecule for attachment of my peptide. So I am telling us now I perform some reactions to attach this peptide on the drug molecule. So we have this OH molecule. This is a linker molecule. This is called maleimide, and this is NHS. The molecule is called NHS maleimide. NHS is a living group, good living group. So with this OH, I react with this molecule, and this is acted as a catalyst. Next I will show you the mechanism. So first, this OH attack here, this carbonyl carbon, and this living group release, and from this this molecule that is modified, I call docetaxel as a DS, and since it's modified to modified DX. Now we have add this peptide to this molecule. I use this maleimide linker because this SH can interact with this double bond of this maleimide. So when I add this molecule with this MDX, so this SH attacks this carbon and it forms this peptide modified docetaxel. Okay, so here I will show you the mechanism. First, this catalyst, this base as I used to deprotonate the hydrogen molecule from OH, and then it becomes negative and it performs the SN2 reaction, and after that this attachment, and then it is just addition reaction of the SH to this maleimide part. Now I prepare another molecule. Okay, so this molecule which I prepare, this is a peptide modified docetaxel, but this molecule has no color. So if I want to trace that molecule inside the cell, means where this, if I add this molecule to cancer cell, where it is going, to locate that molecule, I also prepare another molecule with fluorescent label. Okay, so during synthesis of my peptide, I use this fluorescent molecule to attach with the NH2. If I come to this this part of the peptide synthesis, so with this part NH2, I attach another fluorescent molecule. Then then the complete molecule I use for this docetaxel functionalization, and I got this molecule, this FITC molecule, this fluorescent molecule for this to trace that molecule where inside the cell. So for my biological experiment, I use two cell lines. One is MCF7 cell line, that is a cancer cell line. Another cell line I use HEC290T cell line, that is non-cancerous cell line. Actually, this cell line has not that receptor of the NRP1 receptor. Okay, so this is a cartoon diagram. I'm just showing you that this is the neuropilin 1 NRP1 receptor. MCF7 cell line has this receptor, and this HEC293 cell line do not have this NRP1 receptor. So first, I'm showing you one data that is on MCF7 cell line. So I treated that MCF7 cell line with this FITC PMDX molecule. That is the color molecule where peptide attached docetaxel. So I found that the molecule attached on the cell surface. Okay. So as we see the cartoon diagram, that since the docetaxel, red one is docetaxel, and this is the peptide, so it can interact with the neuropilin 1 receptor. So it can interact with the cell surface, and then it will go to the inside the cancer cell. Okay, but in case of a HEC293 cell line, there is no receptor, so it is not able to interact. Hence this molecule is not able to go into the HEC290 cell through any receptor mediated internalization is not possible in this cell line. So I am just showing you data of the MCF7 cell line. I have also data for this HEC290 cell line that I did not showing you here. I will tell you later. So in the MCF7 cell line, I found that the FITC labeled docetaxel molecule is entering into the cancer cell, but it is a qualitative data. Okay, but we cannot to quantify this data. We perform another experiment, that is a fluorescence activator cell, that is a FACS experiment. For student, I just want to tell you that how this experiment is FACS. What is the FACS experiment? Okay, so in FACS experiment, all these color cells means all the cells are passing through a channel, and there is a laser. So that laser can identify check the each cell whether there is a fluorescence or not. So then we will get signal in computer. If there is a signal, if there is a fluorescence, we will get a signal in computer. Okay, so we studied the cellular uptake process through this FACS experiment. So in FACS experiment, we found that okay, this is the HEC290 cell line. Okay, this treated cell line, and this is also MCF7 cell line, treated cell line. So we found that MCF7 cell line has more signal, more green signal compared to the HEC290 cell line. Okay, so this data indicates that that FITC PMDX molecule entered into this MCF7 cell line much more compared to the HEC290 cell line. Actually, when the molecule we add some molecule to cancer cell, so there is different kind of internalization process. One is receptor mediated internalization, and another type of internalization is also possible that we cannot stop here. So some of this FITC molecule also entered in the HEC290 cell line. That is not receptor mediated. That is some because cells has also other kind of internalization mechanism which is not receptor mediated. Through that mechanism, some molecules entered into this cell that was not able to that internalization was not able to stop through this functionalization. But if we compare the relative internalization of the cancer and the non-cancer cell, we found that molecule entered the cancer cell much more compared to the HEC290 non-cancerous cell. And if we see this bar diagram, this is the treated cell line, green one. So MCF7 has more internalization compared to the HEC290 T cell line. So next part, I perform the cell killing activity study. Actually, this percent viability means how much cells are present after the drug interaction. Okay, so this is called percent viability. 100% viability means all the cells are alive after drug treatment. So when there is no drug, so there is 100% cells are alive. So as the blue one is the docetaxel, red one is the peptide modified docetaxel. As we increase the drug concentration, okay, when I drug concentration, the number of cell killing increases. This viability assay is performed using the MTT molecule. MTT is a molecule which is transferred to another molecule, pharmazine, by the enzyme secreted by the cells. Okay. With this, some reducted enzyme are secreted by the live cell. So as much as the live cell, the amount of enzyme will be more, the more MTT will be converted to pharmazine. That pharmazine has some color that is analyzed by absorption spectroscopy, and from that signal we can calculate this viability. So when there is no cell killing, all the cells are live, so there is all the molecules, all the MTT molecules converted to the all the MTT molecules converted to the pharmazine and giving the color that is the 100% color. Okay, with the 100% signal, the signal we called it 100%, and that is no drug. So as the number of we increase the drug concentration, the number of cancer cell is decreases because the cells are killing by this docetaxel. And as we increase this concentration, the cell killing also increases. So less number of cells are present as we increase the amount of drug. So if we see this 1000 nanomolar concentration, so the number of cells killed by docetaxel is less than the number of cells killed by PMDX. So PMDX has ability to kill more cancer cells. So because of the functionalization of the docetaxel with the peptide molecule, the activity of PMDX is activity of this molecule is much more compared to the docetaxel, as we observe from the cell viability data. So if when we attach a peptide molecule with docetaxel, its activity increase because the internalization of that molecule through the receptor is increased. So next I will show you that this data in the HEC290 cell line, that is the non-cancerous cell line. So in case of non-cancerous cell line, we found that the reverse result. Reverse result means in case of non-cancerous cell line with 100 nanomolar concentration, we found that docetaxel kills the cells much higher, but the PMDX is not able to kill the cancer cells, not able to kill the normal HEC290 cells. So this result suggests that that's entry or the internalization of this PMDX molecule to the cancer cell to the cell occurred through the receptor mediator. Since the HEC290 T cell has no receptor of the NRP1 receptor, no NRP1 receptor or less no NRP1 receptor. So this molecule PMDX is not able to enter to the head-to-near T cell. As a result, head-to-near T cells are not killed and are alive after this drug treatment. So it from this result, from these two results, okay, this result and this result, we can say that this docetaxel, it is a normal chemotherapeutic drug. Okay, this drug can kills also kills the cancer cell as we see here, and also it can kill the normal cell also. Okay, but this is the targeted drug. This PMD is a targeted drug. Docetaxel is modified with the peptide. The targeted drug. This targeted drug is able to kill the cancer cell much more compared to the normal drug, but it cannot kill this normal cell. That is our goal, to make a drug which cannot which will kill the cancer cell but not the normal cell. So this result indicates the targeted way delivery of the drug molecule. Next, I will talk about the activity of the docetaxel and the microtubule. Actually, docetaxel drug interact with the microtubule. What is the microtubule? Microtubule is the cytoskeleton protein of a cell line. Okay, this is the picture of a microtubule network. The blue one is the nucleus. Okay, I take the cell lines and I use Hochest. There is a dye molecule that is called Hochest. With Hochest, I color that nucleus. It will specifically color the nucleus of cell line. Okay, and then I use some antibody that will color antibody and primary antibody that will attach with the microtubule protein, and then secondary and color secondary antibody, red color secondary antibody to this cell line, and it will color the microtubule network. Microtubule cytoskeletal protein of a cell. Okay, but when we add docetaxel molecule, then this docetaxel molecule add on this microtubule. Okay, if we say this docetaxel, so it will add on this microtubule. After adding this docetaxel on the microtubule, the normal activity of the microtubule has a normal activity, that is polymerization, from polymerization and depolymerization through this that is called microtubule dynamics, polymerization and depolymerization. Through this microtubule dynamics, cells is used for the cell division. Okay, in cell division we know different phases like prophase, metaphase, anaphase. Okay, and in some phase the chromosomal segregation occurs. For this chromosomal segregation, this microtubule dynamics is more important for this chromosomal segregation. So if this microtubule dynamics stop, this chromosomal segregation is not possible. Therefore the cell division is stopped when we add this docetaxel molecule. So now, for in our case, we also want to see what happens with this PMDX molecule. So in case of PMDX molecule also, we found that this PMDX molecule also enter into the first molecule into the cancer cell, and it also destroy this microtubule, the good structure of the microtubule. So you can see there is the good structure of the microtubule, but here this microtubule structure is damaged. It's not that like that good structure means you can see the cell size is much higher. Okay, since it is a cytoskeleton protein, when it is damaged, the cell size also decreased. Okay, so microtubule forming like a bundle type of structure. That will indicate that a microtubule network is also affected with this PMDX molecule. And next, I will show you another data, that is the data on this tumors model. Tumor model means first before doing an experiment on mice, we can use some kind of experiment on tumor on a tumor model. Tumor model is we just make a MCF7 cell line on a petri dish and keep it few days. It forms some spheroid. Okay, some spheroid. Spheroid means a lump of cancer cells in a petri dish that we can use that we can treat it. We can say it's a tumor. Okay, now we use the in control. Control means there is no drug treatment, and in another petri we use PMDX, another petri we use docetaxel. So after this treatment, we observe day by day and measure the size of this tumor spheroid, and we found that for this control panel, every day the size increases. For docetaxel that is the green line. For this docetaxel, first few days size increase and then size going to decrease of the tumor. But in case of PMDX, the size is always decreasing. From date means the size did not increase; it all is decrease always. So we can also observe from this data that PMDX molecule, that is the peptide modified docetaxel molecule, is much more effective for cancer treatment compared to this docetaxel. And after this, for another important aspect is the cell death. For when we can develop a drug molecule or we can introduce a new drug molecule, we have to also analyze how this drug killing the cancer cell. There is two kinds of killing. One is apoptotic killing, and this necrosis killing. If we make some molecule and if it kills if its cell killing pathway follow the necrotic pathway, then it causes various kind of inflammation. It making inflammation. Inflammation is not good for our body. So we have to make the drug which can kill the cell, cancer cell, like in a apoptotic way. Apoptotic pathway is a normal cell killing normal cell death pathway. So every day in our body, some cells are cell growth and cell also growing cell growth also occur, and also there is some cells also fall. Are the apoptotic pathway normal cell killing pathway. So if we use, we have to also check whether the drug molecule which we discover or which we synthesize is killing the cells in a apoptotic way or not. Because if it kills the necrotic way, then the drug molecule is not good for our health, because it will make inflammation. Inflammation is not good. So we have to make it in an apoptotic way. Therefore, we have another test, that is test with the NXNV. And what NXNV does? NXNV is a molecule that will, if a cancer cell kill in a apoptotic, if a cell kill in an apoptotic way, then NXNV binds on the cell surface. If we see the cell surface, there is a lipid molecule, and that is another lipid molecule is a phosphatidylserine. Initially, in case of healthy cell, the phosphatidylserine are inside. This part is the inside and this part is the outside. So this part phosphatidylserine, the serine lipid molecule are inside. When cells death occurs to apoptotic way, then some phosphatidylserine molecule come to the outside. When it comes to the outside, then NXNV can bind on the cell surface. This is this way we can detect a cell death pathway, whether it is if it apoptotic way, the NXNV will bind, and we can detect this NXN5 if we use the FITC level NXN5. Then we can detect the presence of NXN5 from our study. I will show you. We can also use this FACS analysis. If we FACS analysis. If we use FITC level NXN5, then FITC level NXN5 will attach on the cell surface. And now, if we pass that cells through the channel, then with the laser we can detect that how many cells are labeled with the NXNV. That we can use from this computer. So we can show you this data, that is apoptotic cell death. That is a FACS analysis of the cell death pathway. There is two coordinate. One is the X coordinate showing the FITC signal, that is the green signal, and for the cell death pathway, another molecule is used, that is the propydium iodide. Propydium iodide is another molecule which will bind on the nucleus and responsible for the red signal. If we go to this slide again, so when we use propydium iodide and NXNV together, so since in necrotic pathway, cell's membrane is completely damaged, so propydium iodide can go inside the cell and bind with the nucleus. NXNV can also bind. Binding of NXNV is less in case of necrotic pathway, but in case of apoptotic pathway, the NXNV also bind on the cell surface, and propydium iodide also can enter and bind with the nucleus. But in case of necrotic pathway, only propydium iodide will goes into the cell, goes into the cell and bind with the nucleus, because there is cell surface damage. So if we use annexin B and propydium iodide, if cell death pathway follow the necrotic pathway, then we will get only the signal from the propydium iodide. But it is apoptotic pathway, we will get signal of annexin B and also propydium iodide. Now, if we see this slide, so this is the x-axis. X-axis showing the signal of the FITC signal of the annexin B, and this y-axis is showing the signal of the propydium iodide. Okay, so here this is the normal. These are the cells not treated with any drug molecule. So it comes to the Q3 quadrant. Q3 quadrant is the reference quadrant, showing that more of the cell line. This each dot indicates each cell line. Okay, so with this study we use huge number of cell line. So each dot indicate cell, each cell, one cell, two cell, like each dot is a cell. So the cells which we are using are the healthy cells or not. To check that, we first do the FACS with this control cell. We found that in control cells are coming in Q3 quadrant, means there is no binding of annexin. We did not add any annexin V or any propydium iodide. This is a control cell without any annexin V and propydium iodide. Then we use the drug molecule. Okay, this is a docetaxel drug. When we use the drug molecule and also add the annexin drug molecule and also add the annexin B and propydium iodide, then we found that cells are coming in Q2 quadrant and also some cells are coming in Q1 quadrant. So, but if we calculate this number of cells in this Q1 and Q2, we found that this Q2 quadrant has most number of cells compared to the Q1. So Q2 quadrant, this quadrant indicates the signal coming from this annexin B, that is FITC signal, and also signal coming from the propydium iodide, this part and this part. So cells are coming in this Q2 quadrant and also some Q4 quadrant. Q4 quadrant also. Q4 quadrant means cells are attached with the annexin V but not attached with the propydium iodide. Q1 is only propydium iodide, no annexin V, and Q2 is annexin V and propydium iodide. So more number of cells in the Q2 quadrant indicates there is a cell with that cells after this drug treatment. Okay, these cells are attached with the propydium iodide and also annexin V. So it also indicates apoptotic pathway. And with our molecule, that is PMDX, we also found more cells in the Q2 quadrant. It also indicates that these cells has attached with that annexin V, that is giving FITC signal, and also propydium iodide. Okay, cell line. So these cells are also killing through the apoptotic pathways. Now, if we compare the number of cells in Q2 quadrant, that is, we found that in case of PMDX, this is more number of cells in the Q2 quadrant, indicates that more apoptotic death occurs when we are using PMDX molecule instead of docetaxel. So the synthesized drug also killing the cells in apoptotic pathway, that is proved through this FACS analysis. Okay, next. So after this part, I have shown that how can we modify a drug molecule with a peptide. Okay, but there is many such complex drug molecules. If we see the structure, complex drug, if we see the structure of the doxorubicin, so in case of doxorubicin, it is not possible to chemically modify the drug molecule. So we can use another type of chemistry to deliver the drug in a targeted way to cancer cells. That is the chemistry using liposome. So liposome is a micelle formation of the lipid molecules. Okay, so when we use the lipid molecule and add some water and sonicate it, it forms a liposome. It has two parts. One part where the one part is like a membrane of lipid, that is and inside this membrane all the long chain of the lipid molecule are arranged. So this part is the hydrophobic part, and inside of the lipid molecule it entraps some water molecules. So it is the aqueous part. So it's a hydrophilic part. So this is a hydrophobic part and this is a hydrophilic part. So based on our drug molecule, so if we add some drug molecule during liposome preparation, it can enter either into the hydrophilic part if it is a hydrophilic drug, or it can enter into the hydrophobic part if it's hydrophobic drug. So if we use docetaxel drug molecule, if we see the structure of the docetaxel, okay. If we see the structure, if in case of docetaxel molecule, it cannot be modified chemically, because if we modify the drug chemically, then the activity may decrease. So we can use that drug itself into the liposome. During liposome preparation, we can use that drug and it will enter into the hydrophilic part. Okay, so water soluble drug can enter into hydrophilic. Now we can modify this lipid molecule. We can modify the lipid molecule. We can make some peptide molecule along with this lipid molecule. Okay, use for liposome preparation. That lipid molecule, that the peptide molecule will act as a target for the cancer cell. Let's say this is the two lipid molecule, POPC and POPC. These are two lipid molecules used for liposome preparation. Now with this lipid molecule, if I use some peptide, one peptide is LDB peptide, and this is YSA peptide. So YSA peptide is targeted for another receptor. So this is EphA2 receptor of cancer cell, and LDB peptide alpha 5 beta 1 receptor of the cancer cell. Okay, so these are the receptor targeted peptide sequence, LDB and YSA. So if we see that this molecule, so this part is a peptide part, and this is the long chain part. This is C16 long chain. Okay, so C16 long chain look like the long chain of the lipid molecule, and this part is a hydrophilic part. Okay, so lipid molecule has two parts. One is the hydrophobic part, this is long chain, and another is the hydrophilic part. So this part acts as a hydrophilic part, and this part, this is a long chain part, is act as a hydrophobic part. So we can use call this type of molecule is a lipopeptide. Okay, since this is a peptide attached with the long chain molecule, we can call it a lipopeptide. So if we use this lipopeptide along with this normal lipid molecule, it will give some liposomes like this. Okay, so this type of lipid, this is the lipid molecule or the normal lipid molecule, and the lipid molecule with peptide will be like this. The hydrophilic part, peptide part, will be exposed outside. So this exposed outside part can also interact with the cancer cell receptor. So this is the preparation of the lipid liposome molecule. Liposome, that is, first we add the lipid molecule in a solvent. Then we can remove that solvent and dry it. Then add the aqueous, which is water. So we can add the drug molecule in this part. So when we add the drug molecule in this part, it may dissolve in water or may not dissolve in water. We can add this drug molecule. No, sorry. If we add if we have hydrophobic drugs, hydrophobic drug, we have to add this drug molecule here when we are giving the lipid molecule inside. So hydrophobic drug are soluble in chloroform. So if we have hydrophobic drug, we can add this hydrophobic drug with this lipid molecule. But if we have hydrophilic drug like docetaxel, we can soluble it water and this or any buffer, or we can use add this on the lipid molecule. So after when we add this buffer on the lipid molecule, the lipid layer is hydrated, and because hydrated means it's water goes inside the lipid, and then if we sonicate it, so after sonication it will give you liposome of multi-layer liposome. Okay, it may have one layer or two layer, that will give you liposome. Okay, now if we can use the TEM using TEM transmission electron microscope, we can see that how liposome looks like. This is the liposome looks like. So this is the structure. This is the these are the liposomes with the normal liposomes with the normal lipids like POPC and POPT. Okay, now I prepare this liposome also, that is with the LDB docetaxel. Okay, docetaxel is a drug that I use during liposome preparation, and LDB is the LDB lipopeptide, this peptide, LDB lipopeptide. So, and also this with YSA lipopeptide, I prepare another liposome, that is YSA docetaxel. And PL means the total. Total means another liposome I prepare, that where there is also YSA and also an LDB. So there are four type of liposome. This is the one type of liposome, that is no targeted liposome. It is not targeted liposome because here I use the peptide, no peptide are used here. only the normal lipid POPC, POPG. This is a one kind of targeted liposome used prepared with the LDB lipopeptide, and also I add some drug molecule docetaxel inside this liposome. This is a YSL. These are the liposome targeted with the YSA peptide, and these are the liposome targeted with both LDP and YSA peptide. So the targeted means that this part YSA LDP part are exposed outside. Now when we use this molecule for the cancer treatment, okay, now we use the cancer cell. A375 is the one kind of cancer cell, and WI38 is another type of cell that is called non-cancerous cell. Okay, so A375 cells has both the receptors for LDP and YSA. So we found from this viability data means cell. Sorry, I'm just putting it. Sorry, there is a phone call is coming. Sorry. Okay, so for this cancer, okay, for this cancer cell line, we found that when for cancer cell line, we found that the control there means there is no drug, means no liposome. So with this, but when we use the drug molecule docetaxel, we use 100 nanomolar docetaxel in all the cases. So when we use the docetaxel at 100 nanomolar, it kills some cancer cell. But when we use the docetaxel in liposomal formulation, like LDP docetaxel, YSA docetaxel, it also kills the cancer cells. But it's slightly much more than the normal docetaxel. But when we use the dual targeted liposome, that's targeted with the LDP and YSA, it kills the more docetaxel, more cancer cells. But in case of normal cells, we found that docetaxel is able to kill the normal cells, but in case of dual targeted, it's not able to kill the normal, not killing the normal cells. So after targeting ability, what happens that the drugs are not killing the normal cells, but it kills only the cancer cells. So that's is the goal of my research. So in summary, I just want to tell you that I have shown the two type of approach of targeted drug delivery. There are always there are many kind of approach. I have shown here on two type of approach. One approach that is a chemical modification of a drug molecule with the peptide, and another approach is a liposomal approach where chemical modification is not possible, because always we cannot modify a drug in a chemically. We can use this liposomal approach to target a cancer cell for cancer treatment. So this is all about my research. Okay, so these are the things. Now you can ask your question and give me your suggestions.
Thank you, sir. So if anyone has any questions, so kindly ask from the audience. Anyone having any questions? Students, any questions? So can I ask one question, sir?
Yes, okay.
So in this liposomes that you have prepared, sir, is there any role of like that for any drug to be uniform? Is there any role of polydispersity index?
Polydispersity? No. You can use any kind of drug for inside the liposome. You can use any kind of, no. It's not in polydispersity index is not important here.
Okay, but how are you ensuring that uniformity is there? Just on the basis of functionality?
No, from the basis of functionality, I cannot say the uniformity. But if you see the data from this data, you can see that if the liposome is not targeted, then it will give the similar laser. It will not give the result means it will not able to kill the cancer cells, right? So if the liposome is not targeted, then it will also give the data like docetaxel or like LDP. Okay, so dual targeted, we can see that these are the killing the more cancer cells. And also I have some more data on like cellular uptake. Okay, from cellular uptake data also we have found that when we are using dual targeted or target or single targeted, are that type of liposome going much more inside the cancer cell compared to the non-targeted liposome. So here, from that point of view, we can say that our targeted delivery approach is the goal is our goal of our research is fulfilled. Okay, during the results, so we did not check the polydispersity of the liposome, but we can check the we check that the size of the liposome from the TM, and also another instrument where we can get that size. We can check that size. That size are almost in nanomolar range, 200 100 nanomolar range. But the polydispersity index, it varies. Some liposomes are very big, some liposomes are not big, small liposomes. So polydispersity index is big, but all the liposomes are targeted or not, that we can understand from the results. Okay, from the cellular uptake or MTT assay, that way we can understand liposomes are functionalized with the peptide.
And this, sir, in this preparation of peptides, is it like similar to Merrifield synthesis?
Yes, Merrifield solid phase synthesis. But for this lipopeptide, we have to first synthesize that peptide part. Then we need to attach with another molecule, that is a succinic anhydride. Okay.
Okay. I
The
get it, sir.
succinic anhydride, we got this functionality, and this molecule was synthesized separately. Okay. This separately, then attach this molecule with this part. Of the peptide.
And then you characterize by M by Z, GCMS.
Yes, it is characterized by mass spectroscopy and also NMR spectroscopy.
Sir, and one another question, sir. Do you for this regression, do you go for McLafferty or this kind of arrangement to know about the M by Z?
No, we use the ESI mass spectroscopy and MALDI.
Okay.
From
MALDI, yeah.
We can get the exact mass. If we have the not get the exact mass, then it is required for this check whether from where the mass is coming. But we got the M by Z marks.
Fragment, yes, sir. Okay. Sir, would you like to ask any question? Sir.
Saurabh, can I make few comments?
Yes, sir. Yes, sir. Sure.
Yes, sir.
Dr. Saha, it's a wonderful presentation. I liked it. You have a strong background in chemistry, and you have presented thorough data around biology. It's very, very enjoyable. I understood all the experiments very well. Nice, lucid presentation. Many, many thanks. Very good.
Sir.
Very good. Keep up the tempo. Well done.
Thank you, sir.
So, if there are not any more questions, so we would conclude this first session for the time being.
Saurabh, one comment regarding the heterogeneity of the dispersion of the materials, drugs in question in the liposomes that you were asking. Would it be possible to check the electron microscopic picture of the liposomes to validate or negate, rule out is duschinta? That could be done, perhaps, no?
Yes. From this
pictures for even distribution of
the liposomes.
By seeing the radii of all these spherical particles, so if the radii is almost similar and it varies within 2%, the error bar. Yes, sir. Okay, sir. Thank you, and for your humble presence. And we would like to again join for the second session, and it will be around. It will be starting from 2 o'clock. So thanks a lot, sir, for your lucid presentation and for enlightening up us about these peptide-based molecules for this cancer treatment and your thorough research, and we are truly grateful. Thank you. We will.
One last comment. This speaker from Ramakrishna Mission's College for undergraduate studies, chemistry. Me also from Ramakrishna Mission College called Vidyamandira, chemistry. And this speaker did his PhD from IICB. I did not know this.
Yes,
I also did my PhD from IICB. When you joined, I just left around that time.
yes, sir. I joined in 2011.
And I retired in 2012. You worked with whom?
I work with Dr. Surajit Ghosh. He is now in IIT.
Surajit,
Yes,
right.
Dr. Surajit Ghosh.
I gave him all my instruments. I can recall.
Yeah.
Right. Very good. Very good. Thank you very much. Keep up the tempo.
Thank you, sir.
Thank
Sir,
Thank you, sir. Thank you. Oh, okay. We are having one question. Yes, you may ask.
Yes, yes.
Sir, which is more suitable and effective, sir, radiotherapy or this drug treatment?
Actually, there are many therapies for cancer. One is surgery, radiotherapy, chemotherapy, target therapy. Now, doctor will decide based on patient's condition. If you have some cancer in some complex area, okay, like brain, okay, there if you use some radiotherapy, or if you have cancer in some complex area, maybe in your lungs, okay, some area where radiotherapy is not possible because it will damage the other. Also side effect. Therefore, the drug therapy comes. So, in targeted therapy, you can target each particular cancer cell. It will not affect the other cell lines. That is the purpose of targeted therapy. So, doctor will decide which therapy is required for which patients based on the patient's condition or other medical backgrounds of also patient is important. So we have many kind of therapy. Doctor will decide which therapy is important for that patients.
Okay, sir. Thank you.
Any questions, or we will conclude this session? Any question from the audience? Okay, I believe there is no question, sir. So thank you. We will be meeting again for the second session, which will be starting at 2 o'clock. So kindly be punctual, and we shall be seeing at 2 o'clock. Thank you, everyone.
Okay, thank you.